Hi again. We're going to get into a few of the basic ideas behind the anatomy and some of the properties that we use to describe the cardiovascular system, specifically the vascular system. So we're going to focus right now on vessels, blood vessels. This should be mostly review, and I encourage you to look at other materials if you have questions, all the way back to the fundamentals course in our department. You can find materials, videos, lots of textbook materials on these topics. So I hope it's review, but I think there's some key themes to really discuss and to make sure that we're clear on. And the first of those is the role of the vascular system in this whole cardiovascular system. So the vascular system solves a challenge, a physiological challenge that all living organisms have, and that is to transport things. It's really as simple as that. So transport of the materials we need to function, the fuel we need for metabolism, we need to remove waste products, byproducts, we need to get oxygen, we need to get rid of CO2. All these things are transport challenges at the end of the day. And so each living system has to find a way to solve that problem. And the solutions depend on the species, not surprisingly. So if we think of very simple organisms, unicellular organisms, the yeast, the bacteria, the amoeba, the paramecium, some of these things that are mobile, that actually have motion, they have mechanical function. They do more than just sit around, being pushed by the streams of liquid that they're in. They have the need for transport just like more complex systems. However, in these systems, that transport happens largely by diffusion. Now, diffusion should be a known concept. If not, please look it up, remind yourself. It's a very important concept. Darcy's law is one explanation of it. It's one of many laws that we will note depend on gradients, as we mentioned in the opening lectures. The gradient of concentration in this case is what drives diffusion. Diffusion is a slow process. Diffusion doesn't transport very far, very fast. And so it has built-in limitations that are not a problem when we have single-cell organisms. There, the size scale is so small that, and the activity level is modest enough that diffusion is a suitable means of transport. However, as we move up to slightly more complex organisms, and here we have the example of the bivalve, the mollusk, so these are clams and similar shellfish, they have a system which does depend on diffusion, and all living systems depend on diffusion. This won't go away. We'll see diffusion in complex organisms and in humans. But in these medium scale or medium complexity and medium size organisms, we start to see a circulatory system emerge. And that circulatory system is characterized by the fact that there's an oxygen carrying molecule and a liquid that can carry nutrients. And all of these materials then get transported through vessels of some sort and through a liquid that transports them. So it's more than diffusion, although diffusion plays a key role. It includes actually some flow of a liquid that one could characterize as blood or like blood or have the function of blood. So this is very true and very important in these middle scale organisms. Now what we see is that the vascular systems are not closed compared to the mammalian systems we're used to. So we see that there's blood leaving a heart-like structure, a structure that pumps this liquid around, but that there's diffusion between the output side of this system and the return side of the system. They could be a sinus, it could be some just nearby spaced vessels, but there are separations, there are gaps between what we would call the arterial side and the venous side of the circulation that we don't see in mammals, which is this third major category that we're going to see again and again throughout this course, and that we hopefully know something about already. And so here the idea is that this is a closed system. The blood that leaves the heart returns to the heart. It may be different blood in terms of what it has on board, but it's still blood, and that blood does not, as a whole, diffuse off into other tissues and become reabsorbed. So diffusion is important. Diffusion is what drives nutrients across the intravascular or the perivascular space. So the space between inside the blood vessels and outside the blood vessels. That is diffusion driven. And once, let's say, ions reach the intracellular space or get inside of cells, then diffusion definitely dictates how they move about within their domains. But there's a high level, high capacity transport system that is driven by blood in this case. And so we're going to now evaluate, or not evaluate, we're going to try now to examine the structural elements of that vascular system. Here's another diagram you'll see again and again. I like to use it a lot because it has all kinds of, it's sort of a mix between a little bit of anatomical and probably a lot schematic. So we see the left heart, the right heart, but we're going to focus on everything sort of to the right of that, or maybe even between the two sides of the heart, between the right side of the heart and the left side of the heart, because that's really where the blood transport happens. And we can see that even more schematically. We can go all the way away from anatomy and truly see this as a schematic situation. This is what this diagram shows, where the right side of the heart is still separate from the left side of the heart, and in between are the lungs directly capturing the blood from the right side and leading it back to the left side. And then all these other systems, all these other anatomical systems, functional systems within the body, and each one of those has its own blood flow. One of the key things to understand here is where we have serial flow and where we have parallel flow. This obviously has a huge impact on the function, on where blood flows, how much blood flows, all these other parameters that are going to become important to us. So it's important to note that the right side of the heart feeds directly into the left side of the heart with, of course, the lungs in between. So it's a serial connection between the right side of the heart, the lungs, and the left side of the heart. In these multiple systems through which blood from the left ventricle flows, these are all in parallel. So the blood is divided up among those systems. And the regulation of those various flow rates is something we'll talk about in some detail. It's an important aspect of physiology. So this is the high-level schematic circuit we can deal with. And we can go back to that original diagram, that sort of schematic / anatomical one, and focus on a key part of it where the actual transport happens versus the delivery. So you can think of this a little bit separately as a system whereby blood is moved, and the major vessels in the vascular system are only there to move blood. So they're transporting blood to other parts of the body, but they're not really doing anything functionally for the body. The fact that an artery is flowing through a tissue does not mean that anything comes from that big artery into that nearby tissue. That's where we have to get into the smaller vessels, and that's where actual delivery happens. So now, if we focus down on the lungs as one example, and then this systemic capillary region as another, this is really where delivery happens. This is where the nutrients transfer from inside the vessels to outside the vessels. This is also where the blood goes from the arterial side, which we think of again as largely oxygenated blood, goes through these capillaries. Oxygen transport takes place in the lungs in one direction, takes place in the tissues in the opposite direction, and then it comes out of those capillaries and flows back into the veins. So again, think transport separate from delivery, and we'll treat each one of these topics functionally differently as well and explain some of the behaviors. So if we focus on the transport part at this point, then we can think we're in the domain of hemodynamics. That's the word you've hopefully come across. This is blood flow inside of tubes. And the simplest approach to understanding how blood flows in tubes is to use what's known as Poiseuille's law. This was a law actually derived by two physiologists around the same time, working together. Hagen was the other one. So they developed this relationship, this more or less simple law that is incredibly powerful and helpful for us to explain how blood flows. So we've talked about properties of blood flow in a previous lecture, the resistance to blood flow, the compliance of blood vessels. Here's where this takes on some quantitative form and some hopefully understanding that comes from that quantitative form. So Poiseuille's law, you can see written down below here, is a function of pressure differences. So once again, we're back in the same territory where gradients matter. In this case, the gradient of pressure. Blood flows from high pressure downhill to low pressure. And that's what we see in the diagram, and that's what we see in the equation. The difference between the pressures at either end of the piece of vessel that we're examining, that determines how much drive there is, the driving force that's pushing blood through that segment of vessel. The other term in the equation represents the resistance of that or to that blood flow, or its inverse, depending on how you want to picture it. So what we see here is the relationship embedded in this resistance term that includes the things we would expect it to include if we kind of think a little bit about the mechanics of the problem here. Obviously, if we have a larger area, a larger surface area, then we would generally expect there to be a lower resistance. If we have a greater length overall, we would expect there to be a higher resistance. Similarly, if we have a higher viscosity or thickness of the blood, we would expect a higher resistance. And if you look at those terms in that equation, the pi a to the fourth over the 8 nabla L, that's the inverse of resistance. So now we have an expression we can simplify as the flow is equal to the difference in pressure divided by the resistance of that blood vessel segment. This looks like Ohm's law, feels like Ohm's law, functions like Ohm's law, and is one of the many laws that we'll come across that have this form, this apparently linear form that relates a flux or a flow quantity with a driving force, a gradient quantity. This is just, again, one example we'll see of that again and again throughout this course. So this is the one that determines our blood flow, and it's an important one for us to know, but we can apply that and actually derive numerical answers or numerical approximations for blood flow using that expression, provided we can figure out the parameters, provided we can determine reasonable values for all the parameters that go into the equation. Now, the second major element, property of blood flow that we have to take into account is compliance. So compliance is derived really from the notion of stretch, elastic recoil. We can think of it coming from initially a rubber band is a one-dimensional example. We pull on that rubber band, we let go of the force, the rubber band comes back again. If we take that same concept and apply it to a volume surrounded by a surface, then that material in the surface, like a balloon, has those same stretch characteristics, that same elastic recoil, and that's what leads to compliance of vessels, where the recoil or the elastic deformation of the vessel happens in the radial direction in general. So we have flow along the vessel, and then perpendicular to that flow direction, we have distensibility of the vessel, and that we call compliance, and that allows us to even quantify that relationship, because it's really a relationship between the pressure applied to that segment of vessel and the resulting volume that that vessel segment takes on. Now, these are important properties that describe elements of our vascular system, hence we have to talk about them here. And we see a plot like this one that captures the compliance of blood vessels, and the typical picture that you'll see in these diagrams is pressure will be along the x-axis and the blood volume, relative or absolute, somehow will be along the y-axis. And we see here two separate curves. We see the arterial curve and the venous curve. And we can also now describe the parameter of compliance in terms of the numerical relationship between volume and pressure. It's literally defined as a change in volume associated with the change in pressure. And we, of course, quickly recognize that that is the same as the slope in this particular rendering of the data. So if we examine the slope and how the slope changes, then we can come to some conclusions about compliance and how compliance changes with pressure, and whether or not it's linear, for example. Is it a linear function of pressure? And we'll talk about that in some of the discussion sessions. Now, this combination of resistance and compliance allows us to put together pretty realistic models of of vascular flow, of the flow in the blood supply. But one of the questions we have to ask ourselves is what determines the resistance, that's cross-sectional area, what determines the cross-sectional area of a vessel, and even more subtly, what determines the compliance of a vessel? Are all vessels have the same compliance? The plot would suggest otherwise. We have different slopes, dramatically different slopes, for the arterial compliance compared to the venous compliance. So obviously there's something different about these vessels. And how do we explain that? Well, as many times in this course, to explain this functional difference, we have to look at structural differences. And if we look at the makeup of vessels and compare them across the different segments, let's say leading from the heart, the large arteries that direct the blood from the heart out into the systemic circulation, let's say, then we see that there are a number of differences along that progression. Here we see a plot, for example, that describes everything from the aorta, the arteries, veins, and the vena cava. So it takes us through the entire blood supply, skipping over the capillaries, which we'll get to in a sec. In this plot, we see the diameter, the diameter and the radius change as we move from these large vessels near the heart to smaller vessels, even small arteries and veins that are further away from the heart. So that's going to change the cross-sectional area, and that will clearly change resistance in ways that we can even quantify. The compliance is also different. You could say, why is the compliance different? Well, there are different materials that make up the walls of those vessels. It's like having a balloon with a very thin wall compared to a balloon with a very thick wall. Those balloons will have different compliance. And so we can see that when we just look very simply at the makeup of the vessel walls in terms of four components that we typically see in these vessels. And the endothelium is that innermost layer we see in all vessels, and all vessels have that layer, typically just one layer, one endothelial layer. But everything after that, it can be quite different. There's different amounts of elastic tissue. There's different amounts of fibrous tissue. Elastic tissue makes something recoil more strongly. It's a thicker elastic band if it has more elastic tissue. The fibrous tissue gives it some stiffness. So a vessel with more fibrous tissue will be a stiffer vessel. And that hopefully also makes sense. And finally, the smooth muscle. The smooth muscle has passive characteristics that are somewhat elastic, but more important in the context of regulation is that that muscle can be activated. So we can have constriction of the vessels coming from stimulated smooth muscle. And that becomes an important topic we'll get to shortly. Now, if we dive into the smaller vessels, the arterioles, capillaries, and venules, we see we've jumped into a whole different unit system. On the left, the units were in millimeters. Now we're down into microns or micrometers. And we see they're much, much smaller vessels and also have very simple structures. The capillary is the simplest of all. It's literally just a layer, an endothelial layer. And that makes some sense when we go back to think about its function, and that is the function of delivering. That's where the ions have to cross, let's say, from the blood into the interstitial space, or gases have to transport back the other way, the CO2 that gets produced during exercise. There has to be transport possible at the capillaries for them to have that function of being the delivery agents. And so, indeed, that is where we see the thinnest walls, just in endothelial layers. The arterioles, venules are different, have certainly more structure, but again are quite different. And we notice, for example, that the arterioles have smooth muscle, which means they can be regulated, whereas the venules really can't. And that becomes an important element of very local regulation of blood flow. The arterioles are largely responsible for that by the mechanism of stimulation of that smooth muscle, which alters the cross-sectional area, the radius of the vessel, and hence changes its resistance. And again, we'll get into all that. So this structural differences determine functional differences. We say this again and again in physiology, and it's certainly true here as well. Here we have a diagram that actually links the different vessel types with their function in broad strokes. You know, the aorta is not there to deliver anything, it's just there to transport the blood. But it does have a function in terms of it dampens a very pulsatile pressure that comes from the heart and starts to smooth out that pressure. It does that because it has elasticity, it has some recoil, it can smooth out those very abrupt pressure fluctuations that occur inside the heart. And each of those different segments of the vasculature has a different function. And you see the capillaries is there for exchange. That's another way of saying that's where things are delivered. That's the the delivery or the exchange of ions or other molecules across the wall from inside to outside and back again. Again, structure, function, relationship. I told you we'd see that again and again. Now we have to think a little bit about another structural functional relationship, and that has to do with the way blood flows, the hemodynamics, at a qualitative level at this point. If we had a vascular system that was purely resistive, if our vascular system looked more or less like our plumbing systems at home, then we would see some interesting characteristics. Whenever the anything changes in such a system, the blood flow changes, the output changes, the pressure changes, and that change is reflected immediately through the entire system. In a perfectly, completely resistive system, this is how things would function. Of course, that's not how our vascular system works. It's not really completely how our plumbing at home works. If you look carefully enough, you'll see there are absorbent elements to the system that provide some compliance, that it provides some expansion of the liquid in the system and smooth out some of the fluctuations we get when we turn on a tap suddenly. So when we have flow through resistive vessels, we just describe it with Poiseuille's law, and we can write down equations. We can determine the resistance of everything. We have a very simple system. We have the equivalent of a circuit that consists of a power supply and only resistors. And we know that's a nice, simple circuit. We can actually analyze that, hopefully. And we see the pressures change stepwise as we go through different resistive elements through that system. The key thing here is that time is not a factor, in the sense that the minute something changes in one part of the system, that change is more or less immediately reflected throughout the rest of the system. Now, in compliant vessels, that's different. In compliant vessels, we have some time delay. The stretching, the elastic recoil, that builds a time delay into our system. And so we have a situation where we could have a steady state blood flow situation where the inflow is equal to the outflow. But then if for some reason, let's say the heart contracts and the inflow to that artery increases, so the flow in the Q1, as we have it in this diagram, increases compared to what it was before, then the Q2 doesn't immediately increase to that same level. Instead, the vessel, if it's compliant, has the ability to absorb some of that volume that's flowing into that segment of vessel. That's the nature of compliant vessels. And so we have a time delay, we have a transient state when there's some distension. That distension has limits. The limits are defined by the compliance. And we get a new steady state that arises, in which eventually the inflow and the outflow are the same, unless the inflow changes again. So if we have a heartbeat, there's an increased inflow that lasts for a short period of time, and then it goes down again. And so we have a constant fluctuating situation of flow and a constant pulsatile behavior in our vessels. Pulsatile behavior that we can actually measure. This is what a pulse measurement is actually capturing. This is pulsatile pressure waves that are changing the way our vessels also distend. And so we have a more or less well-defined sequence of events that we can pull apart and even apply equations to capture that behavior. And there's a separate video I would encourage you to have a look at if you're in any doubt whatsoever about how these quantitative approaches work and how we end up with a first-order differential equation that we can then solve using numerical methods. Please do watch that video. I really can't tell from your backgrounds whether this is going to be brand new material or something you recognize. It does actually come from the Fundamentals 2 course, so you may have seen it before if you've taken that class. But please have a look at that if you're unsure about anything. So in summary, the main points here are that flow in vessels, so this is the transport of vessel of blood to different regions in our vascular system, occurs as a function or is determined by the key properties of the system that we capture in the resistance and the compliance. So we have to know what those are. We just have to be able to talk in those terms. Those are the properties of interest and of relevance here. Resistance causes instantaneous changes. So a change in resistance causes an instantaneous change in flow. If we have a resistive system, a change in inflow causes an immediate change in outflow. All these things happen instantaneously, whereas when we have compliant vessels, as we do throughout our living systems, we have a time course to take into account. So we have to solve, in the end, we have to solve typically ordinary, but some form of differential equations that have a time-dependent aspect to them. That adds some complexity, but we can handle that. We're engineers. Compliance is rarely linear. So if you look back to the graphs of the vasculature, the arteries and the veins, and you first of all notice that they're quite different. So they have different slopes, which means they have different compliance. And they also have largely nonlinear in the sense that over the broad range of pressures, the slopes are not constant. So that's hopefully, a key element of linearity is how, when you plot the two parameters, how they relate to each other. Are they a straight line or do they form a curve? And you can see those graphs of compliance are curved, whereas the graphs of resistance, there we have, again, flow as a function of pressure in a purely resistive system. Those tend to be linear relationships over a broader range. But again, linearity is not perfect there. This is the challenge of quantifying a cardiovascular system, is that oftentimes we'll use equations in a more qualitative sense than you may be used to. If we go deep enough into the details and gather enough information and are complex enough in our models and the underlying math, we can solve these problems in a quantitative way. And you'll see examples of that in some of the tools that we'll use for homework assignments. But you should appreciate the challenge that's involved in that. This is a non-trivial situation, non-trivial circuit to solve. And then finally, again, arteries and veins respond quite differently. They have different structures, so they have different functions. And that function difference is reflected in different parameters, different properties. So they have different resistances depending on their cross-sectional area. And they have, more importantly, different compliances, which are a function of the structure, of the makeup of the vessel wall. So that will hopefully give you enough information about the vascular system, both to tackle the lab this week, and then as a background for what we're going to talk about next time, or next week, which is the control of this entire system. How do we determine how those parallel branches that we described earlier, different parts of the vascular system, they get different amounts of blood flow, how do we regulate that? How does the body regulate that? What are the factors that drive it? What are the mechanisms that allow it to happen? This is mostly the structural and the property perspective that'll give us the tools, the working concepts with which to get into these explanations of regulation and all the control systems that we have to talk about. So be sure these concepts are clear to you, comfortable with you. If not, bring them to the discussion session, talk about them amongst yourselves, review some of the material that I've got on the Canvas page, and we'll look forward to seeing you again soon. Thanks so much for your attention.